Now we go for the next question. The type of pseudopodium of Entamoeba histolytica. Now in the life history of Entamoeba histolytica, one stage we call it as trophozoite. Now as we observe the trophozoite, it appears to be amoeba. The body is covered and protected by plasmalemma, and this is advanced anterior end, and this is the posterior end. The entire body is covered by plasmalemma. Below plasmalemma, what is present? Ectoplasm. Below ectoplasm, what is present? The endoplasm. At advanced anterior end, what is present? The pseudopodium is present. How many pseudopodia are present? Only one. If one pseudopodium is present, it is called monopodial. What type of pseudopodium it is? The lobopodium. What is meant by lobopodium? The pseudopodium is blunt and finger-like. Such type of pseudopodium we call it as the lobopodium. Here the pseudopodium helps in the food collection and amoeboid movement. Now, what type of pseudopodium of Entamoeba histolytica? One is myxopodium, second one phylopodium, third one exopodium, the last one is lobopodium. The correct answer is lobopodium. Now we go for the next question. Number of daughter cells formed from metacystic form of Entamoeba histolytica. Now, as we observe this part, we call it as the lumen of small intestine. This one we call it as the lumen of small intestine. Whenever the human being takes the contaminated food and water, along the contaminated food and water, what stage enters into the body of the man? That is tetranucleated cyst. How many nuclei are present? 4 nuclei. Now, one tetranucleated cyst passing different parts of alimentary canal and enters into the cavity of small intestine. Now, in the cavity of small intestine, as we observe, this is a tetranucleated cyst. In the cavity of small intestine, what is present? The pancreatic juice. In the pancreatic juice, what is present? The trypsin. Now, the trypsin dissolves the cyst wall. The process of dissolving cyst wall, we call it as existment. What is the site of existment? The lumen of small intestine. Now, after existment, this tetranucleated stage will come out. Now, it is called metacyst. Now, it is called metacyst. Now, in the life history of Entamoeba, where we have to observe 4 nuclei. One is tetranucleated cyst and the second one we call it as metacyst. Now, the metacyst undergo reproduction. First nucleus divides, karyokinesis. What is meant by karyokinesis? Division of nucleus. How the nucleus divides? By a process called mitosis. Karyokinesis followed by cytokinesis. Now, one metacyst gives rise to how many daughter individuals? 8 daughter individuals. These 8 daughter individuals are unicellular, made by one cell. In each daughter individual, how many nuclei? Only 1 nuclei. Now, based on this information, here the question. The number of daughter cells formed from the metacystic form, 8 daughter individuals. Later, they become the mature trophozoites. Now, what is the correct answer for this question is the first option, that is 8. Now, go for the next question. Match the following. Now, Ronald Ross, second one Charles Leveron, third one Camillo Golgi, fourth one Grazi, and the fifth one we call it as Granham. Based on this information, the student immediately get an idea regarding the what type of parasite, that is the Plasmodium. Now, as we observe the list 2, French army doctor. The second one we call it as Italian scientist. Third one we call it as Italian professor. Fourth one we call it as zoologist. And the fifth one we call it as monograph on Plasmodium. Now we have to discuss one by one. Sir Ronald Ross, he is British army doctor. He worked at Secunderabad. On one day, he identified the Wucista Plasmodium on stomach wall of female Anopheles mosquito. So, Sir Ronald Ross associated with the Wucist. What about the Laveron? His complete name is Charles Laveron. Charles Laveron, French army doctor. He first identified the Plasmodium in the blood of malarial patient. He was considered as the father of malariology. What about the Golgi, Italian scientist? He explained Plasmodium present in the RBC. He explained the erythrocytic cycle. It is also called Golgi cycle. What about the Grassi, Italian professor? He explained the life cycle of Plasmodium vivax in the female Anopheles mosquito. What about the Granham? The complete monograph of Plasmodium given by Granham. Based on this information, what is the correct answer for this question is? That is third option. Now we move for the next question. Assertion. Sporozoite exhibits the wriggling movement. Now assertion. The sporozoite exhibit the wriggling movement. Now in the life history of Plasmodium, what is the infective stage to the man? What is the infective stage to the man? That we call it as the sporozoite. The sporozoite is infective stage to man. The anterior posterior ends are pointed. The middle part is swollen. What is the length of the sporozoite? 15 microns. What about the width of body? That is 1 micron. Now the entire body is covered and protected by elastic pellicle. Now below pellicle what is present? Plasma lemma. Below plasma lemma what is present? Ectoplasm. Below ectoplasm what is present? Endoplasm. Now as we observe the body wall of sporozoite, what are present? The longitudinal microtubules. What are present? The longitudinal microtubules. Because of the longitudinal microtubules, the sporozoite performs what type of movement? The wriggling movement. Now, assertion. Sporozoite exhibit wriggling movement. What is the second one? The pellicle contains microtubules. How the microtubules are arranged longitudinally. The entire body is covered and protected by elastic pellicle that is secreted by plasma lemma. In the pellicle, the microtubules present. Because of the contraction and relaxation of microtubules, the sporozoite performs wriggling movement. The microtubules are made by what type of protein? That we call it as tubulin protein. Now, based on this information, what is the correct answer? Both are correct or explains A. Now we move for the next question. Assertion. In the life history of Plasmodium, man is considered as the secondary host for the Plasmodium. A reason. A sexual phase of Plasmodium is completed in man. Now as we observe the life history of Plasmodium, in the life history, the primary host, this symbol indicates primary and this symbol indicates the secondary. What is the primary host? The female Anopheles mosquito. Because in the female Anopheles mosquito, the parasite completes the sexual cycle. What about the secondary host? The man. In the universe, this is the only exception. In the life history of Plasmodium, the man is secondary host. but in all remaining parasites the man is the primary host. What is meant by primary host? In the host body, sexual cycle is going to complete or sexual stages are present. Now in the life history of Plasmodium, the man is secondary host because in the man body Plasmodium completes what type of cycle? Asexual cycle. The asexual cycle is brought by multiple fission. What is the other name of multiple fission? Schizogony. Now based on this information, what is the correct answer? Both are correct or explains A. Now we move for the next question. Following stages can attack RBC to start Golgi cycle. Again the question is repeating. What are the stages going to attack RBC to start Golgi cycle? Now, as we observe the Golgi cycle is also called erythrocytic cycle. It is also called erythrocytic cycle. In the Golgi cycle, we have to take the RBC. What are the infective stages to RBC in the Golgi cycle? One is the cryptozoite and the second one is micrometacryptozoite and the third one we call it as erythrocytic merozoite and the fourth one we call it as hypnozoite, in brackets activated. All these are the correct options for this question. What are the infective stages to RBC? The cryptozoite, that is the first generation of merozoite, attacks RBC. Micrometacryptozoite, second generation of merozoite, attacks RBC. Erythrocytic merozoite, third generation of merozoite, attacks RBC. What about the hypnozoite? The hypnozoite is in the dormant condition. Whenever it is reactivated, the hypnozoite starts the Golgi cycle. Now all these options correct. Now based on this information, what are the following stages can attack the RBC to start Golgi cycle? One is cryptozoite, that is correct, first generation of merozoite. Micrometacryptozoites, second generation of merozoites. Erythrocytic merozoites, fourth one all. All these options are correct. Now go for the next question. Arrange the sequence of the following in the ascending order in the life cycle of Plasmodium. Now, what are the stages given? One we call it as cryptozoite. Second one we call it as signet ring stage. Third one we call it as amoeboid stage. And the fourth one we call it as schizond. Now based on this information, we are getting one idea, that is the Golgi cycle. Now as we observe the Golgi cycle, in the Golgi cycle, what is the infective stage to RBC? Cryptozoite, micrometacryptozoite, erythrocytic merozoite or reactivated hypnozoite. Now one is the cryptozoite. Now the cryptozoite becomes the trophozoite. Now the trophozoite becomes the signet ring stage. The signet ring stage becomes the amoeboid stage. The amoeboid stage becomes the schizond. Finally, we are getting erythrocytic merozoites. This is a brief regarding the Golgi cycle. In the Golgi cycle, the infective stage to RBC cryptozoite. First it becomes trophozoite. Next it becomes a signet ring stage, followed by amoeboid stage, followed by schizond, followed by erythrocytic merozoite. Now you have to observe. One is cryptozoite. Now this is. The second one is signet ring stage. And the third one is amoeboid stage. And the fourth one is schizond. This is the order of stages in the Golgi cycle. Now based on this information, the correct answer is the first one. Now we go for the next question. Read the following statements. Now the first one is hypertrophy of infected RBC during erythrocytic life cycle of Plasmodium occurs during amoeboid stage. Now as we observe the Golgi cycle, in the Golgi cycle, this is the signet ring stage. In the signet ring stage, this one we call it as P means parasite. The parasite appears to be a ring-like, and this is the nucleus. N means nucleus. Now the nucleus appears to be a diamond. Now it is called the signet ring stage. Why the parasite appears to be a ring? Because inside the parasite body what is present? A vacuole is present. Now the signet ring stage is followed by amoeboid stage. As we observe the amoeboid stage, no doubt it is infected RBC. Now as we observe the vacuole, slowly the vacuole disappears. Now the nucleus comes to the center and the parasite develops the pseudopodia. Now the parasite appears to be amoeba. In the amoeboid stage, as we observe the RBC enlarges in its size. So in the amoeboid stage we have to explain hypertrophy. In which stage the infected RBC enlarges? In the amoeboid stage. So the first statement, hypertrophy of infected RBC during the erythrocytic life cycle of Plasmodium occurs during amoeboid stage. It is correct. Second one, Schaffner's dots are present in the cytoplasm of RBC. Now in the amoeboid stage, the parasite feeds on hemoglobin. Now the parasite releases red colored antigens. The red colored antigens are called Schaffner's dots. Where Schaffner's dots are present? In the cytoplasm of infected RBC. Where we are going to observe the Schaffner's dots? In the amoeboid stage. Where RBC enlarges? In the amoeboid stage. So the second statement, Schaffner's dots are present in the cytoplasm of RBC. The second statement also correct. What about the third one? Hemojoin is produced from the hemojoin. Now in the RBC, as we observe in the RBC, what is present? Hemoglobin. This is iron and this is the protein. Now the protein eaten by the parasite. Now this is leftover. It is not eaten. It is called heme. The heme becomes hematin. Finally the hematin becomes the poisonous hemojoin granules. Whenever the hemojoin granules are mixed with blood plasma, the patient gets the symptoms of the malaria. Now the iron part is not eaten by parasite. This is the heme converted into hematin. It finally converted into hemojoin granules. Now what is the third statement? The hemojoin granules are produced from hematin. Now based on this information, all these statements are correct. What is the correct answer for this question is the fourth one. Now go for the next question. Match the following. Now the list one, signet ring stage. Third one, erythrocytic schizogony. It is also called Golgi cycle. Third one, gametocytes. And the fourth one, hemojoin granules. And the next one we call it as merozoites. They are called first generation merozoites, second generation merozoites, third generation of merozoites. Now go for the list 2. Sexually dimorphic stages. Second one 48 hours. Third one malarial symptoms. Fourth one early trophozoite vacuole. And the fifth one erythrocytic schizogony. Now based on this information, as we observe the signet ring stage, in the signet ring stage, in the parasite body, a vacuole present, but it has no contraction and relaxations. Now because of the presence of vacuole, The signet ring stage is also called early trophozoite vacuole. Now, what about the erythrocytic schizogony? Erythrocytic schizogony is also called Golgi cycle. Golgi cycle completes in 2 days. That means 48 hours. Now, gametocytes. What are the gametocytes? They are sexual stages. Male gametocyte, female gametocyte. What is the infective stage to the female Anopheles mosquito? Gametocytes. So, the gametocytes are sexually dimorphic. One is male, one is female. Hemogyne granules. After completing the Golgi cycle, we are getting the poisonous hemogyne granules. Whenever the hemogyne granules are mixed with blood plasma, immediately the patient gets the symptoms of malaria. Now, what about the merozoites? As we observe the merozoites, they are called erythrocytic schizogony. That means, what are the merozoites? They may be cryptozoite or micrometacryptozoite or the erythrocytic merozoite. All these merozoites going to attack RBC. Now, that cycle we call it as Golgi cycle or erythrocytic schizogony. Based on this information, the answer for this question is the second one. Now go for the next question. Assertion. Gametocytes alone can withstand the effects of the digestive fluids in the stomach of mosquito. Again repeating, gametocytes can alone withstand the effect of digestive fluid in the stomach of mosquito. A reason, the pH of digestive fluid is favorable for further development of gametocytes. Now, as we observe the Golgi cycle, after completing the Golgi cycle, we are getting erythrocytic merozoites. No doubt, the erythrocytic merozoites repeatedly attack RBC. That process is going on. After some time, the erythrocytic merozoite enters into a fresh RBC and becomes the gametocyte. The gametocytes are classified into the male gametocyte and female gametocyte. Now, the gametocytes, what is the lifespan of gametocyte? That is 7 days or 1 week. Within this period, the gametocytes must enter into the body of female Anopheles mosquito, because in our body the pH is more, the body temperature is more. These two factors are not suitable for the gametocytes to undergo further development. But in the body of female Anopheles mosquito, it is invertebrate. So, in the body of female Anopheles mosquito, the body temperature is less, body fluids pH is less. The less pH, the less body temperature, favorable for the gametocytes to undergo further development. That is called sexual cycle. Now, based on this information, assertion, gametocytes can alone withstand the effect of the digestive fluid in the stomach of mosquito. That is correct, because in the female Anopheles mosquito body, all stages are going to perish except gametocytes, because they are highly resistant. They can withstand to the contents of the female Anopheles mosquito. The reason, the pH of digestive fluid is favorable for further development of gametocytes, because in the body of female Anopheles mosquito, the temperature is low, the pH is low. Automatically, a sudden change in the pH, a sudden change in the temperature, the gametocytes are going to start the sexual cycle or Ross cycle. Now, based on this information, both are correct or he explains A. Now, the answer is, now we go for the next question. Assertion. Plasmodium exhibits alternation of generations. What is the other name of alternation of generations? That is metagenesis. The other name we call it as metagenesis. Now, as we observe the life history of Plasmodium, here one host is the female Anopheles mosquito, and the secondary host is man. Now in the man body, the Plasmodium completes the asexual cycle. In the female Anopheles mosquito body, it completes the sexual cycle. After completing the sexual cycle, we are getting the sporozoite. After completing the sexual cycle, we are getting the sporozoite. Now the sporozoite enters into the man body and completes the asexual cycle, that is pre-erythrocytic cycle, exoerythrocytic cycle, and Golgi cycle. After completing the Golgi cycle, finally we are getting the gametocides. Now the gametocides enter into the body of female Anopheles mosquito and complete the sexual cycle. That means asexual sexual cycles alter with different hosts, because female Anopheles mosquito is invertebrate and it is vertebrate. Here sexual, here asexual. Alternation of asexual sexual cycles with alternation of hosts. So that the Plasmodium is the best example for the digenetic parasite. Here assertion, the Plasmodium exhibit alternation of generations, correct. Reason, asexual cycle of Plasmodium in man alters with the sexual cycle in mosquito. So what is the correct answer? Both are correct or explains A. Now we go for the next question. Read the following statements. Diploid motile stages in the life cycle of Plasmodium is oconite. You have to follow the statement once again. First statement, diploid motile. Now as we observe the life history of Plasmodium, what are the diploid stages? One we call it as zygote, and the second one we call it as oconite, and the third one we call it as oocyst. No doubt all these are the diploid stages. But what is the difference? The zygote, it is non-motile. And what about the oconite? It is motile. What about the oocyst? It is also non-motile. All these are the diploid stages. Now here what is the first statement? The diploid motile stage. Here what is the diploid motile stage? Oconite. The diploid motile stage in the life cycle of Plasmodium is oconite. The statement is correct. Second one, first nuclear division in the oocyst is a reductional division according to the Bano. Now as we observe, this is the stomach wall of mosquito, and this one we call it as the basement membrane. SW means the stomach wall of mosquito, and this is the basement membrane. Below basement membrane, what is present? The encysted oconite. The encysted oconite we call it as oocyst. Now as we observe, encysted oconite, that is called oocyst. Now inside the cyst what is present? Oconite. The oconite is diploid. According to the Bano, during the sporogamy, what is the first division? The meiosis 1. What is the other name of meiosis 1? Reduction division. Because of the meiosis 1, the diploid number of chromosomes reduced to haploid. Now statement 2 is correct. Third, time taken for the completion of sexual cycle in mosquito body, 10 to 24 days. The third statement is also correct. Now what is the correct answer? All are correct. Next question. Read the following statement about the Plasmodium. One is anisogamy. What is meant by anisogamy? The fusion of dissimilar gametes. Now this one we call it as the ovum. The ovum is round and non-motile, and this is the fertilization cone. In the fertilization cone, what is present? Cytoplasm and the nucleus. Now this is the male gamete. This is the male gamete. The body of male gamete is the flagella-like. The fusion of dissimilar gametes we call it as anisogamy. Because of the anisogamy we are getting the diploid stage that we call it as a zygote. Now the first statement, anisogamy result in the formation of zygote. The first option is correct. Second, the cyst wall of oocyst is secreted by oconite. Now this one we call it as the stomach wall of mosquito, and this one we call it as the basement membrane. This is called the basement membrane. The first one is called stomach wall. It is lined by basement membrane. Below basement membrane the oconite settles. Now the oconite undergoes encystment. It develops a cyst around its body. Now this one we call it as cyst. In the cyst how many layers are present? 2 layers. Now one layer is derived from the stomach wall of mosquito. The second layer secreted by oconite itself. Now the second statement, the cyst wall of oocyst. In the oocyst how many layers are present? 2. One is derived from the stomach wall of mosquito. One is secreted by oconite itself. So the second statement also correct. What is the third one? Motile zygote is called oconite. What are the diploid stages? Zygote, oconite, oocyst. Zygote non-motile, oocyst non-motile, oconite is the motile. So here the given 3 statements are correct. What is the correct answer for this question is all statements are correct. Now go for the next question. The details of life cycle of Plasmodium vivax in female Anopheles mosquito was clearly given by. Now as we observe the introductory part of Plasmodium, the Plasmodium first identified by Charles Laveran. Name the person explained Plasmodium present in the RBC, Comilo Golgi. Name the person identified the oocyst, Sir Ronald Ross. What about the Grazi? He explained the life cycle of Plasmodium vivax in female Anopheles mosquito. The monograph of Plasmodium is given by Granham. Now here, what is the question? The life cycle of Plasmodium vivax in female Anopheles mosquito explained by Grazi. What about the Golgi? He identified or he explained Plasmodium in the RBC of man. What about the Ross? He identified the oocyst on the stomach wall of mosquito. What about the Short? The Short and Granham, they stated that the sporozoite lives in the blood plasma just only for the 30 minutes. That statement is given by Short and Granham. Now based on this information, what is the answer for this question? That is Grazi. Now go for the next question. Gametocyte of Plasmodium developing. Now after completing the Golgi cycle, we are getting erythrocytic merozoite. Now the erythrocytic merozoite enters into a fresh RBC and become the gametocyte. Now the male gametocyte present in one RBC, the female gametocyte present in one RBC. Where the gametocytes mature? Where the gametocytes mature? One is the spleen and bone marrow. In the bone marrow and spleen, the gametocytes mature. The spleen and bone marrow produces RBC. Inside RBC, the gametocyte present. Now, the gametocyte of Plasmodium developing. One is liver cells, second RBC, third WBC, fourth salivary glands of mosquito. Now what is the correct answer? Based on this information, that is the RBC. Now we go for the next question. The process of formation of microgametes from the flagellated body. Now as we observe the microgametocyte, in the beginning as we observe the microgametocyte is converted into microgametes. How many microgametes? The microgametes number is 8. Initially, the male gametes or microgametes are fused. Later, the microgametes separated from the male gametocyte. The process of separation of male gametes from the male gametocyte, that we call it as exflagellation. What about the gametogeny? The formation of gametes from the gametocytes, that we call it as gametogeny. Now, the process of formation of microgametes from the flagellated body, one is spermatogenesis, the second one spermiogenesis, third one exflagellation, fourth one oogenesis. The correct answer for this question is exflagellation. Now we go for the next question. Substance responsible for raising body temperature and feeling chillness in malaria. Now after completing the Golgi cycle, we are getting the poisonous hemojoin granules. As we observe the poisonous hemojoin granules, whenever they are mixed with blood plasma, we are getting the symptoms of malaria. One symptom is cold stage, one symptom is hot stage, one symptom is the sweat stage. Now based on this information, one is hemojoin granules, Schaffner's dots, quinine, amidine. What is the correct answer for this question? That is hemojoin granules. Now we move for the next question. As we observe the next question, read the following statements. Scolex is the pear-shaped structure. In the life history of Taenia solium, the first one we call it as, this is scolex. At the tip of scolex, what is present? The rostellum. Now on the dorsal side one sucker, ventral side one sucker, lateral side two suckers. Now the shape of scolex is pear shape. Now the first statement, scolex is pear shaped structure, correct statement. Second one, the lower row of hooks are larger than upper rows. Now as we observe the rostellum, these are upper row hooks and these are lower row hooks. How the hooks are arranged? The hooks are arranged alternatively in how many rows? 2 rows. The hooks of outer row larger than that of the hooks of inner row. But here what is the statement? Second one, the lower row hooks are larger than. That is the wrong statement. Third one, the hooks. How many hooks are present? 20 to 32. They are made by what type of chemical material? Chitin. So the third one, hooks are made up of chitin. Now out of these 3 statements, the first one and the third one is correct. Now we go for the next question. Assertion. The neck is considered as the zone of proliferation. Now as we observe, this part is scolex and this is the neck. This part is neck, this is the posterior end. Now as we observe the posterior end of neck region, this posterior end performs asexual reproduction. With the help of asexual reproduction, the neck region gives rise to what type of part? That we call it as strobila. So that the neck is considered as the zone of proliferation. The strobila is proliferated, so the A is correct. Or new segments are formed from neck region. Now In the strobila, what are present? Proglatids. The proglatids are formed from the anterior end. As new segments added from the anterior end, that is called pseudometamerism. Based on this information, both are correct and R explains A. Now the next question. Who type of Taenia solium is surrounded by? As we observe the female reproductive system of Taenia, this part we call it as the bilobed ovary, and this part we call it as isthmus, and this one we call it as oviduct, and this part we call it as O type. What is the shape of O type? Round in shape. It is the site of fertilization. The oviduct opens into O type. The vitelline duct opens into O type, and uterus opens into O type, and unicellular Mahalis glands open into O type. What is the function of Mahalis glands? Mahalis glands secretions help to pass the capsules from the O type into the uterus. Now, regarding the question, O type of Taenia solium is surrounded by unicellular Mahalis glands. The answer is the first one. Now move for the next question. Hexacanth larva is developed from. As we observe the development of Taenia, this one is called zygote. The zygote divides into, this is megamere and embryonic cell. The embryonic cell divides into the mesomeres and micromeres. The micromeres divide and become the morula. The morula becomes the hexacanth embryo. So, hexacanth embryo is derived from what type of cells? The micromeres. Now, hexacanth larva is developed from what type of cells? That is micromeres. Now, the next one. List one Rennetti cell, second one nerve ring, third one stylet, fourth one microfilaria, a fifth one darkly stained mass. List 2 rudimentary elementary canal, infective stage 2 mosquito, excretion, pharynx, future mouth. All these are going to observe in the life history of Wuchereria. Now, Rennetti cell concerned with excretion. What about the nerve ring? Present around the pharynx. What about the stylet? In the future, there the mouth develops. What about the microfilaria larva? Infective stage 2, the Culex mosquito. What about the darkly stained mass that represent the rudimentary elementary canal? Now we move for the next question. Read the following stages. One we call it as microfilaria larva. Second one midgut of mosquito. Third one hemocoel. Fourth one thoracic muscles. And the next one we call it as sausage larva. Now this is the female Culex body. Now this is the midgut. In the body of female Culex mosquito, this is microfilaria larva. Initially microfilaria larva present in the midgut. Now the microfilaria larva enters into hemocoel. Later it enters into thoracic muscles. In the thoracic muscles, it becomes the short, stumpy, saucer-shaped sausage. Now, based on this information, arrange in a sequence. Microfilaria larva, midgut of mosquito, comes into the hemocoel, enters thoracic muscles, and becomes sausage larva. Now, the last but one question is, the larval stage formed by second molting. In the mosquito body, two moltings takes place. Sausage undergoes first molting, getting the second stage. The second stage undergoes second molting. We are getting the third elongated infective stage. It is infective stage to man. Now, what is the stage obtained after the second molting? That is the third stage of larva. Now, the last question regarding this topic is, male Wuchereria is characterized by, as we observe the male Wuchereria, the posterior end is pointed and curved. This is cloacal aperture. Near cloacal aperture, unequal copulatory spicules are present. Now, what are the identification characters? The male posterior end is pointed and curved. Cloacal aperture present. One pair of unequal copulatory spicules. Now, in the next class, we have to discuss the remaining part.